Hey mate, last night I woke up and I sound like a British Aussie person. I don't know what happened to me. I think some genes are being expressed that are making me speak like a Brit. That's not true. I just unexpressed it right now. So we're gonna talk about gene expression real fast again. So gene expression, like we talked in the last video, in part one, you see where it's part two. We talked about gene expression being the method that our bodies use to express certain genes, and the expression just means transcribing it first and then translating it. Now we can talk about the three remaining gene expression techniques used by our cells. First is gene dose. Gene dose is just referring to the fact that if we just duplicate certain genes in our chromosomes, that obviously it's going to be transcribed more. What I mean by that is, look, if we have an entire chromosome that we duplicate, we amplify it, of course, now the genetic information on that chromosome will be translated twice, so they're at the rate at least. So the translation rate is twice as fast as before, so it's being expressed at a higher rate. And likewise, if we were to delete those sequences, then that is being reduced, and that is reducing the gene expression right there. That's one way of controlling gene expression right there, gene dose. So we could either copy it or delete it. That's all I'm trying to say. This is an example of copy number variation. We talked about that in mutation. Imprinting. Imprinting is the process by which one allele of a gene is expressed. What I'm trying to say by that is, if let's say that there's a gene inside me that is only expressed from my dad's side, and that means that only allele that's being obviously passed on from my dad to me is being expressed, and my mom's allele is there but it's not being expressed. That is called imprinting, and that really is achieved by non-coding RNAs that are binding to the DNA, and also DNA methylation and histone modification. So you might still be confused on what is imprinting. Imprinting is like I said, is just one allele of a gene being expressed, but this is just for the organism himself. So when, let's say in the example that I gave, when I have kids, my kids don't necessarily have to have the same gene that's being expressed or silenced. So, and this is achieved by the fact that, well, you can see the answer over here. Where would you expect these imprinting to happen or be established? Of course, it's in the germline cell, because in the germline cell we have eggs and we have sperm that is going to be there. We'll talk about this later on, but there's variation when these germ cells are made. This other thing we want to talk about is X chromosome inactivation. You will be pretty much shocked because this got me as well. It's very interesting. So we know that females have, they have two X chromosomes, and one of them is inactive and the other is active. The inactive is also labeled as XI, and the active is labeled as an XA. And that's also kind of made the colors so you can see the difference over here. But if we let's talk more about this real fast. So in human beings, the X inactivation will happen in a very early developing stage in the blastocyte formation, actually. So every cell has the ability to randomly decide that they're going to inactivate one of the X chromosomes. So only one X chromosome is expressed in the cell. This is very cool because if you think about it, males only have one X chromosome, and this is the active X chromosome. So it's important to understand that one of them is active and one of them is inactive, and so what that means is that one cell is using all its genetic code is coming from either the parental X chromosome or the maternal X chromosome from the mom. Obviously, I said maternal again. But here's a question to ask that will help us understand, will help you understand, and of course me as well, will help us understand the structure. So which would the XI, which is a reference to the inactive chromosome, or the XA, which is the reference to the active chromosome, which one would you expect to be, which would you expect, which would you expect to be the heterochromatin? And that's a reference to structure. Remember, heterochromatin means that these chromosomes are dense in nature. They're kind of like tightly packed up together. And that's to avoid transcription from happening. And which has a higher level of DNA methylation? What do you think? Of course, the inactive. The inactive is going to have more heterochromatin. It's going to be in heterochromatin to avoid all these transcriptional enzymes to have access to his DNA and hence transcribe it. And this is the blastocyte right here, and we're going to talk about this in future videos. So the takeaway from this video really should be gene dose is the method of either, just copying the gene, making more copies of a gene, or deleting copies to control expression. Imprinting, one allele of a gene is expressed, and this can be changed as, and this is only applying to that individual as he has kids or she has kids. This imprinting can change. Therefore, it's an epigenetic. It's just modifying the DNA. And we have X chromosome inactivation. Really just to point out that one X chromosome is utilized by the cell, each cell. And there are variations, and it's random and how the cell decides it. So we have a lot of cells in the blastocyte, obviously, right over here. And each of these will decide, hey, look, we want to use the parental or we want to use the maternal X chromosome.